So today we're going to be talking about lipids, but before we dive into lipids, let's talk about the other macromolecules we've discussed so far. So the first macromolecule we talked about were proteins. Proteins are made up of individual amino acids. These are the monomers of proteins. And a protein, the amino acids have an amino group, a central carbon that has an R group coming off of it, and they end with this carboxyl group here. And individual amino acids are joined together in a condensation, and the resulting bond that forms is called a peptide bond. And when several amino acids join together, we call this an oligopeptide, and if it's bigger, we call it a peptide or a polypeptide. We also talked about nucleic acids, and the individual monomer for nucleic acids is a nucleotide. And here I'm showing deoxyribonucleic acid, because we have only the hydroxyl group at the 3' carbon. And nucleotides are joined together by a reaction, a condensation reaction between this 3' hydroxyl and a 5' phosphate. And this forms a phosphodiester bond in the growing polymer. We also talked about carbohydrates, and the individual carbohydrates all have the same molecular formula, CH2O, and they all have lots of hydroxyl groups and a carbonyl group. And here I'm showing a carbohydrate in a ring form, and it's a simple sugar, just 1 unit, so we call it a monosaccharide. That's the basic monomer for a carbohydrate. And these can link together with other carbohydrates via a glycosidic linkage to form a long polymer, something like starch or glycogen. And so you can look at this and start to think about how you want to organize your notes for this class and think about the different functions we've talked about for each of these macromolecules. But today we're going to be talking about lipids. Okay, so this is our last macromolecule. And lipid is a catch-all term for an amphipathic molecule. So what does this mean? This means that there's both a water-loving area to this lipid and a water-fearing area. And so this lipid is a triglyceride, which we'll talk about in a little bit. But what an amphipathic molecule has is it has a relatively small polar hydrophilic region. So you can see there are carbon-oxygen bonds here, and these are polar bonds, polar covalent bonds, and that makes this part of the molecule attract water. The rest of the molecule is primarily made up of hydrocarbons. This part of the molecule contains only carbon and hydrogen. That's why it gets the name hydrocarbons. And the bonds are nonpolar because carbon and hydrogen share their electrons evenly. And because the bonds are nonpolar, this part of the molecule is hydrophobic. Okay, so here's a lipid, and we're going to talk about the different classes. But importantly, lipids do not form a polymer, right? So unlike the other macromolecules we talked about, lipids are a little bit different. So we're going to focus on the 3 most important types of lipids found in cells. These are fats, phospholipids, and steroids. And as I talk about each of these today, it might be helpful to organize your thoughts in a table like the one below, where you can point out the polar part of the lipid, the nonpolar region, so what are the molecules that make up that nonpolar region, and most importantly, the role that lipid plays in a cell or in an organism. What I want to just talk about is the specific structure of a fatty acid. So a fatty acid is a carbon-hydrogen has a carbon-hydrogen backbone or tail, right? So it's a hydrocarbon, and it has anywhere from 14 to 20 carbons, right, on average. So this can be a little bit longer or a little bit shorter, and at the end, it has a carboxyl group. So carbon with its double bond to oxygen, and then the OH bound to that carbon. And it can come in 2 flavors. It could come in this saturated form, in which there are no double bonds between the carbons. So this is relatively straight. It's saturated with hydrogens, okay? So every carbon has its 2 hydrogen, okay, saturated. It can also come in this unsaturated form, in which the carbons can have a double bond with each other, okay? And they can have 1 to many. So you can have a polyunsaturated fat or just have a single unsaturated fat. And so this is unsaturated because this carbon only has one hydrogen and this carbon only has one hydrogen. And these saturated and unsaturated fatty acids have different properties. And so the saturated fatty acid, right, is you can think of it as being very stiff. And because of that, it's solid at room temperature. And this is what we think of as butter or beeswax, right, where you have this long saturated hydrocarbon tail. And unsaturated fats that have this double bond between the carbons are more fluid, okay? And because of that, they have, they're liquid at room temperature, and these are what we have in the kitchen that are oils. And one thing that makes it kind of a little bit easier to remember is that, for me, is that plants like to make unsaturated fats. So if you think of the plants really making unsaturated fats and olives being plants, you can think of olive oil being an oil and that it's unsaturated. So there's a chemical procedure that takes things that are liquid and makes them solid. And this can be useful for preservation or for if you want to do a specific cooking technique, right, that you're dealing with a solid as opposed to a liquid. And this procedure is called hydrogenation. Essentially, so what they do is they break this double bond here, and then they put in hydrogens. Okay, so that's why it's called hydrogenation. And this turns it from a liquid to a solid. But when it does this, and it makes that bond a single bond, it doesn't make it in the right orientation. So instead of being a normal cis bond, it actually makes a trans bond. And our body can't break down those trans bonds. And so this is what's called a trans fat, and this is why trans fat's not good for you, because your body can't break it down. It's a foreign type of bond, and our enzymes just don't recognize it the same way. So here are some pictures of what an unsaturated would look like. Fat in the kitchen, and here's our oil, and then we have our saturated fats, which are solids, so beeswax and butter, and then something like Crisco that makes something that's liquid into a solid by the process of hydrogenation is a trans fat. And so we'll talk a little bit about these structures in class. Okay, so we have our hydrocarbons, we have our isoprenoids, which are the branched, they have those methyl groups off of that hydrocarbon backbone, and then we have our fatty acid, which is just the hydrocarbons, anywhere from 14 to 20 carbons, and then the carboxyl group at the end. Now we can add 3 fatty acids to a glycerol molecule, and this gives us a fat. This is what a fat is, and it's linked, these fatty acids are linked by an ester linkage, okay? This doesn't make it a polymer, but it does make it a complex molecule with different parts, okay? So I want you to know that there's this glycerol molecule that's somewhat polar, and then you have your non-polar hydrocarbon tails of fatty acids. And this molecule, a fat, is also called a triglyceride, okay? Because it's triglyceride. And the primary role of fats or triglycerides is for energy storage. So once we've used up our sugar, right, our glucose, or we've used up the glucose that's been stored as glycogen, then we can go into our fat stores and use that for energy. And so the reason we use that next is because the fat kind of has to be converted into a sugar-like molecule before we can use it to make energy. And so that's the primary role of fat, but fat also serves to cushion our organs. So in adipose tissue that surrounds some of our organs, this kind of protects everything on the inside so we don't damage organs. So the next lipid we'll talk about are the phospholipids. So let's start here at the glycerol part of the molecule. The glycerol part of the molecule is joined by 2 fatty acid tails, and those are non-polar and the glycerol is polar. And then on top of the glycerol molecule is joined a phosphate group. This is where they get the name phospholipids, and an organic group, something like choline, for example. And this makes up the polar head group of this phospholipid. And the one I'm showing you here with the fatty acid tails is usually found in animal cells and bacterial cells. But something with an isoprenoid tail, so a little bit of a different structure than a fatty acid, is something that you'd find in archaea, for example, so another class of living organisms. And you'll find these phospholipids in the cell membrane of those cells. So this is going to make up the membrane of the cell, right? What kind of separates the outside from the inside. And we represent, sometimes instead of drawing this out, we represent this whole region here, this organic group, this phosphate group, and this glycerol as this circle for the polar region. Okay, so this is our polar head, and then these fatty acids are just drawn as squiggly lines for the two fatty acid tails. And these essentially are drawn the same way. But often when you see a membrane drawn, you see this circle that represents all the polar groups and then the tails. So I want to just take a minute to talk about what phospholipids do before I move on to the next lipid, which is a steroid. And what they do is that they form membranes. So because they're ambipathic, because they have these polar regions with these nonpolar tails, when they are placed in water, they do some pretty unique things. And that's that the heads kind of cluster on the outside to face the water, and the tails interact with one another to prevent their interaction with water because they're hydrophobic. And so when they do this, when you drop them in water like this, they form these lipid micelles, right? So it's just one layer. But they can also form, depending on how many lipids or how you alter your conditions, lipid bilayers. And so in the lipid bilayers, as opposed to forming this tiny little vesicle where all the lipids are interacting with each other, they form this double membrane, okay? And so here, these lipids, you have a group of lipids in which their heads are facing the outside and their tails are facing another set of tails from lipids or from phospholipids in which these ones are facing this, if this is water and maybe this is inside the cell. And so they create this membrane where the phospholipid tails that are hydrophobic interact with each other and not any sort of aqueous environment, and then the heads are facing the aqueous environment. And we'll talk more about phospholipid bilayers in the next class. But these phospholipids in the bilayer, right, they can move laterally. Okay, so here they are. There's some fluidity, and we'll talk about factors that influence how well these lipids can move laterally. But they rarely flip to the opposite side of the membrane in healthy cells. Okay, there's also asymmetry, so certain lipids like to face the outside, the external environment, and certain lipids like to face the internal environment. So what gives a phospholipid its specificity? It's that organic group, okay? And so some like outside and some like inside. But when a cell begins to die, that asymmetry is lost. So things that are normally on the inside might flip to the outside. And so that's kind of what phospholipids do, right? And so you're minimizing those hydrophobic interactions and maximizing your hydrophilic interactions. And then the last category of lipids we're going to talk about are steroids. And steroids are a large family of lipids, and they're distinguished by the following things. They have this hydroxyl group here, that's that functional group that makes things polar, and then they have this characteristic bulky 4-ring structure. So here's our rings, 1, 2, 3, 4. So it's pretty obvious when you're looking at a steroid because of the rings. And then it has this isoprenoid tail, right, that branched lipid. And these steroids function as hormones, sex hormones like estrogen and testosterone, but there's another type of lipid called cholesterol, and this is what cholesterol would look like. And in animal cells, it's actually in the cell membrane and contributes to that fluidity. So after this video, and when you come to class, you should be able to explain kind of the structure of each of those 3 types of lipids for saturated and unsaturated fats, right? Whether or not it has all single bonds in the carbon and it's saturated fat and it's solid, or unsaturated fats and you've got a double bond and it's liquid at room temperature. You should also be able to explain phospholipids and steroids and what those look like and what their function is inside the cell. And looking ahead, as I kind of alluded to, we're going to talk about how phospholipids make up the plasma membrane, right? And the plasma membrane is really important, and we're spending a lot of time on it because it's what separates the outside from the inside. And the membrane has to function to keep damaging materials out, allow materials that are needed from the cell in, and facilitate chemical reactions. So that's all I have for today's podcast, and I look forward to seeing everyone in class. Thanks. Bye.